Hey, my name is Rakaya,
I'm Adrian.
and this is our soil discovery project. So for our introduction, we kind of talk about like how understanding the microscopic world is one of the hardest challenges in science, and a lot of the things that we can't see with our naked eye directs us. We don't understand it until it directs us effectively. It's not a word. In the class, we use escape pathogens to narrow down and find a specific pathogen for further investigation and to analyze it and it's resistant to various antibiotics. So we and then to compare this to the real world and other healthcare settings. So for the methods we collected the soil sample, we did serial dilutions um, to obtain the isolated colonies. The master plates were isolated from different colonies and placed onto two new plates, street plates, and then bacteria isolates with inhibition zones, which is over here, and then master plates for antibiotic producers. And then we selected a single isolate and tested it for susceptibility of antibiotics. We did PCR tests to prepare the analysis, and then we completed a whole bunch of tests over here, which we'll go in depth about. I'm finishing the steps, and then we're gonna go through it. And then we have the gel stuff, which is over there. And yeah, so
Okay, so I'll start first. So, first things we did was we had our 2 gram-positive and gram-negative master plates. We found out which, put this under a light to see which colonies had inhibition zones, and we picked 4 colonies that had inhibition zones. So we picked colony 10, 16, 18, and 20. So we further ended up picking 10 to finish the rest of our project. So this is isolate 10 under the microscope. So after we did the microscope, we ended up testing for, we did our PCR, which is right here. And then after that, we tested for our antibiotic resistance. So our bacteria is resistant to ampicillin and penicillin, and the rest are susceptible except trimethylphen, which it was intermediate to. So after we did that, we put it on this, and it had a inhibition zone around S, which is consistent with our gram-positive bacteria.
that's also the sample too, which is the sample of our bacteria. So it being resistant to that was what we were looking for.
Yes. And then after that, I'll explain this one bit. So after that, we did our fermentation test, our biochemical test, but specifically fermentation. So our PID and our MIO, so our PID was negative. Our MIO, which is motility, indole, orethine. So motility was positive, indole was negative, and orethine was positive. So also for our fermentation test, we did a glucose fermentation test, sucrose and lactose. So glucose is bright yellow, so that means it was positive, and it also had no gas buildup. Our sucrose was red, turning yellow with more time. It was red at the time, but like if we would have given it like a day or two, it would have turned yellow, which makes it positive. And our lactose was negative because it just stayed the same color. And we had no gas buildup in any of our tubes.
Also, I did want to make like a quick note about our escape pathogen and what this means. So for the escape pathogen, there's 2 types of things. There's a safe relative and an escape pathogen. The escape pathogen, they come off to be a little bit more infectious in comparison to the safe relative. So we had those 2, which you'll see in the picture, and we were supposed to put the safe relative on.
Yeah, we didn't do that.
But that's okay. It's alright. That's alright. Also, what do I want to say? Oh, these things. So we tested our colonies on 5 different types of agars. We had the slant agar, which was gram-positive. The STA, which is gram-positive as well, which is this one. And then the MSA, which is this one, was gram-negative. PEA, which is this one, gram-positive. MAC, gram-negative. And then EMB was also gram-negative.
So yeah, in our discussion, we figured out that blast results show how. We actually have to go over our blast results. So sorry, backtrack. So our blast results. So we did our blast results, and our bacteria came out to the gram-positive Basilicus hominis, which is consistent with our findings because it's gram-positive. And also, he said that our thing is like a mix of gram-positive and gram-negative
due to the presence of alcohol.
Yeah.
Also, with our bacteria, it had a 88.6 0.76% identity, which means that it's that close to the other bacteria, which is not 100%, but it's still like very close, close enough to consider it. Yeah. And then it also had a 77% query cover, which kind of supports that.
So our discussion, our blast report showed that our DNA sequence is close, it matches closely to the Basilicus genus, which is what we wanted, and it includes, including Basilicus hominis and Basilicus mobilis. So this matches, the matches have a high significance, which means that it's by, the similarity is not by chance, however the percent identity is only around 88.76%. Yeah, about 77% query covered. And
in this experiment, the gram-positive bacterium showed more growth than the gram-negative, which suggests that the antibiotic produced by the Bacillus simplex was less effective, and it was more effective against the gram.
Our microscopic observations.
No, yeah.
Okay. It showed chain-like cells, so this, which is consistent with gram-negative. And we have the dot-like ones that's consistent with gram-positive. So it's consistent with our findings that our colony is gram-positive.
Yeah, with a little bit of gram-negative.
Yeah, with a little bit gram-negative. And yeah, so.
Oh, our sources are
on the back.
right over here.
Yeah. And
what else? So for our next steps, we're basically gonna create a whole new experiment, so to say. So we're gonna test the antibiotic producing bacillus against both positive and negative to see whether it works like with different bacteria. We're also gonna test the amounts of bacteria in antibiotic producing bacteria to see if it actually leads to bigger zones or stronger effects. We also thought about changing the temperature and the humidity and just like the
conditions.
The conditions of the soil. And we also wanted to look up to see whether it's actually stronger against one pathogen compared to others, since that could show if it's more selective on what it kills.
Yes. So thank you for listening. This is our project. Yeah. Bye.